mention one thing, update from yesterday. So I'm not encouraging any patients to take clenbuterol because it's not FDA approved, and that's an obstacle and it's a reason not. So wait for future clinical trials. Secondly, we did show benefits of what's available, albuterol, that is FDA approved, but there's a shortage, many know. And I'm not sure if there's a role for patient advocacy with the manufacturer to work on that shortage. That's just a suggestion. But let me continue quickly with the talk. This is about our phase 1 clinical trial of gene therapy in adults with Pompe disease. Let me see if we get this to advance. Here are my disclosures. Basically shows that I work with companies and also that I do have an interest in this company that's supporting our clinical trial as an inventor. So this is the most important feature, the most important slide from my presentation. And this is the strategy for our gene therapy, which is an AAV8. It's a modified virus. It doesn't produce any disease by itself, but it's modified to carry a gene to make GAA to the liver, and to make the GAA only in liver. So there are two features of that. One is that because the GAA is made at a high level, it's secreted into blood, and the blood carries it to all the muscles where it can be taken up to correct GAA deficiency and to lower the glycogen content. So that's how it treats Pompe disease, and it's like a continuous ERT without all the ups and downs. More on that in a second. The second feature is that by making the protein only in liver, there's a well-known immune mechanism that prevents the antibody response against GAA. So you've heard before with ERT that antibodies are formed against the GAA, in some patients at high levels, and they prevent the benefit from ERT. So this gene therapy suppresses those antibodies, and it can actually make ERT work better potentially in a patient who has already formed antibodies. And at any rate, it won't provoke antibodies, so this gene therapy could work well and have a sustained benefit. Also just mentioned here, we have orphan drug designation, so we could have an approved therapy someday, and that should help the development of the drug. So, oops. This next slide is a comparison then of our results from our research in the laboratory with this gene therapy and with what's known about ERT, which is standard of care. With regard to stability, that's how long the GAA is around. So it's well known that the GAA is high briefly following an infusion of ERT, and then it drops quickly. So there's a short half-life. With this gene therapy, there's a continuous GAA production that goes into the bloodstream and can be taken up by the muscle. With regard to delivery, we know that the receptor is low in skeletal muscle, so the uptake of GAA from ERT is not optimal. Perhaps with this continuous delivery with gene therapy, all of those receptors that are available can continue to bind the GAA and take it up into muscle for increased delivery. I mentioned the immune responses, and in some patients, especially CRIM-negative infants with Pompe disease, there's a high titer antibody response. And at least in our experiments with our knockout mice that do make high antibodies in response to ERT, this gene therapy suppresses those antibodies. So there's an advantage. Given the ability to give a single dose, this could be available to patients who don't tolerate ERT. There could be a potentially larger patient population than there is for ERT. That's speculation. We've seen in our experiments again that there's a more complete correction of GAA deficiency and glycogen storage in a direct comparison with ERT. And this graph shows nicely the single administration with a prolonged effect as opposed to the cyclical and frequent infusions with ERT. So our experimental data, and this is an experiment that we published 2 years ago with a direct comparison of ERT and a low-dose gene therapy. We compared with mice that received no treatment. We went just a month because after 4 treatments with ERT, the mice formed antibodies and start to have allergic reactions. So that's about as long as we can compare the ERT to gene therapy conveniently. And then we looked at correction of the skeletal muscle in these mice with Pompe disease. So what did we see? This is just picture showing the design of 4 weekly injections of ERT versus 1 dose of the AAV vector. And here's the publication for your reference. If we look at GAA in this graph across different tissues, we can see a nice elevation in liver, either for the ERT shown in the middle or for the gene therapy. Now we see that the gene therapy is bumping up the GAA in heart, diaphragm, and quad, but it didn't pass a statistical test. However, we know the glycogen is more sensitive, so we can show biochemical correction more often by measuring glycogen content across these muscles. And so both methods, the ERT and this low-dose gene therapy, lowered glycogen content significantly to a great extent in the heart. We know the heart is most responsive to this kind of GAA replacement. In the diaphragm, too, that's mentioned frequently, it's a very important target. We saw a similar effect from this month, 4 doses of ERT, and from a single dose of this low-dose gene therapy. In the quad, we have a hint here that the glycogen content is going down. And actually, if we look at males only, we saw a significant effect for the gene therapy that we didn't see with ERT. We can look at a comparison then of what's going on with GAA during ERT or with gene therapy in our experiment. So the first thing is activity in the blood, and we can measure a significant and stable elevation of GAA activity in the plasma following AAV vector administration or the gene therapy. And not surprisingly, the ERT didn't elevate GAA activity, and we know again that it has a short half-life. So looking a week later after the last dose, ERT is undetectable in the plasma compartment of the blood. As I mentioned, the antibodies are formed in response to ERT, and this is a strong signal for antibodies that would prevent a good effect from ERT in the long term. And we didn't see those as predicted when we gave the AAV vector. So that was a demonstration of immune tolerance to GAA. That's very important. One feature I want to remember to mention is that in another experiment, we found that if the mice had already formed the antibodies to ERT, we could give gene therapy later to suppress those antibodies. And this is giving a little higher dose, actually a fairly standard dose known in the field, 2 × 10⁻12 vector genomes per kilogram. And here we could see GAA elevation from gene therapy across the board in comparison with untreated mice, so the black versus the gray bars, just a simple comparison of gene therapy to no treatment. And a very nice reduction of glycogen, stored glycogen, across these muscles, more than 50% in every muscle that was studied. And this is a long-term experiment over 36 weeks. So this says that at a little higher dose, we get a very substantial correction of Pompe disease changes in the muscle. Now, our clinical trial is a phase 1 clinical trial mainly designed to look at safety. And we've had 2 groups of 3 patients enrolled at a low and a higher dose. The way we're judging safety is based on carefully monitoring for adverse events or side effects, and especially serious adverse events, and monitoring for clinical laboratory abnormalities. Secondary endpoints, things that we'll look at in addition to look for benefit, include these standard validated endpoints, the 6-minute walk test, pulmonary function testing, especially FVC. Then we do a muscle biopsy before gene therapy and after. So we measure the GAA activity and the glycogen in the muscle. And we also monitor the GAA in serum like we did in our mouse experiment. We'll monitor anti-GAA antibodies and the urine biomarker. The inclusion criteria, so a confirmed diagnosis of Pompe disease based on low GAA in the blood or a skin fibroblast assay, and then confirmation by gene sequencing showing 2 variants that are known to cause Pompe disease. This first study is only for adults, and patients should have been receiving ERT for over 2 years and on a stable dose for over 1 year, so they're very stably treated. They shouldn't have additional benefits from this ongoing ERT, so improvement could be attributed to gene therapy. Exclusion criteria are that the patient can't have a anti-AAV8 neutralizing antibody above 1 to 5. If the patient had any elevation of this antibody against AAV8, gene therapy wouldn't work. That's the current problem that we face, and this has to be addressed later. But for this phase 1 study, then we had to carefully screen patients to make sure they didn't have an elevated anti-AAV8, and about half of them were eligible based upon this. And then as well, if the patient wasn't able to complete the evaluations by different measures, then we wouldn't enroll them in this study. This is a 1-year study with a 4-year follow-up. The vector is given within 4 weeks of the screening visit, and this is very important because we want to make sure there's no time for the patient to get exposed to AAV and maybe form antibodies. And we also check the antibody right when the AAV is given to make sure that it's not present and isn't interfering with the benefit. Everyone needs prednisone prophylaxis, and this is an immune suppression that's critical to prevent known T-cell responses against this AAV8 vector. This is known from previous clinical trials. If you give an affected dose of AAV8, the patient will form an immune response that will eliminate it from the body. And this prednisone prophylaxis is a known way to prevent that, and it's critical to preserving the potential benefit. As I mentioned, the muscle biopsy is done before and after gene therapy, and then we do ongoing muscle and pulmonary function tests along the way. I'm not going to show you those data today, but I am going to note that this is very analogous to a successful clinical trial in hemophilia B that gave an AAV8 vector to produce factor IX for that disease. And those patients in a high-dose group had a benefit for over 3 years. We expect this could be long-lasting for our trial if it works the same, because we're using a very similar design. I mentioned about the need for immune suppression, and that comes from this study too, because these patients, given the higher dose, had a rise in the liver enzyme in the ALT weeks 7 to 10, and that could be suppressed by giving prednisolone, essentially the same drug that we're using. So we've adopted that to preserve any potential benefit. Here's the team. So the lead clinical investigator is Eddie Smith, who's a neuromuscular specialist, and he does the needle muscle biopsies. Many of you have met him before. And he makes independent decisions for the clinical trial, independent of those of us who designed it and are leading the research. So that's an important feature to prevent any conflict of interest. Laura Case, you've met too, who's the physical therapist and helps us with the muscle testing. And my colleague Priya Krishnani is critically involved in this study to support us and to help with her expertise. It's supported by our clinical GSD laboratory, which is a unique resource for measuring GAA and glycogen in a clinical laboratory that's CAP and CLIA certified. And then we have a regulatory team to communicate with FDA from Duke. So this is the only data I can share today. So this is from the first cohort, although we've enrolled both cohorts. And I can say that all subjects are male. They're all middle-aged, average height and weight, and that we've seen safety through week 24. So there have been no adverse events of grade 3 or higher that would be considered severe. I'll mention too that each of these patients has elevated GAA activity in their serum in comparison with baseline. Sorry, my mouth's a little dry. Phase 1 study to determine safety and bioactivity with this AAV8 vector in adult patients with late-onset Pompe disease. And to date, we've seen both of those features. We're looking at secondary endpoints that I'm not sharing today, but this does demonstrate initial safety. And I want to acknowledge funding and resources, especially from NIAMS, who funded this study initially, and then AskBio, who's provided manufacturing and ongoing support. Thank you.